want to introduce our speaker, Angela Zuelow, PhD student at Hatfield. She's going to be talking obviously about winners and losers of Oregon kelps, kind of, this very diverse ecosystem out there, kind of who's doing well, who's struggling with these changes and our uncertain future. And I'll get out of the way here and let Angela take the stage.
Okay, hi everybody. Thank you for having me. I'm excited to be here. So I'm just gonna get straight into it. So for my talk outline today, I'm gonna be going into just a quick personal intro on myself and how I got into loving algae and kelp. Then I'm gonna give you some basics around what are the differences between algae and kelp, and I'm gonna use those terms a lot. And then I'll give a quick ecology intro related to my work, and then I'll talk about where our kelp forests are right now, especially in Oregon, and I'll talk about how my research relates to it, and then I'll talk about some future support and where things are going. So for some quick background on myself, I am from Northern California, and I did my undergrad at Humboldt State, which is now Cal Poly Humboldt, which is the most northern CSU in California. I did major in marine biology, and I tend to get the question, how did I decide to major in marine biology? And it was kind of like on a whim. I liked science when I was in high school, and Humboldt State has a really good marine science program, very similar to OSU. They have their own marine lab and they have their own research vessels. And it was a state school, so very affordable. And I was like, that sounds like a good major. I'm going to do that. And I ended up loving it, so that's good. And then for my research as an undergrad, I was at Humboldt between 2014 and 2018. And if you have been around the oceans at all around 2014, between 2014 and 2018, we had a huge sea star wasting event and we lost a lot of our stars. That upper right picture, you can see that sea star I'm holding has dropped two of its legs and will eventually completely waste away. So there was a lot of money being thrown at that issue at that time. So that's kind of what a lot of people were focused on. So I studied sea stars and their wasting. And then I also was focused on their armor. So specifically piezaster, which is very common here in Oregon too. They have this like outer structure spines out here, which not all sea stars have that. And we were studying to see how that differed based on the habitat they were in. So did they beef up their spines if they were more wave exposed, things like that. So definitely focused a lot on sea stars as an undergrad. But I did do a summer internship, and I actually did it here at Hatfield in Newport. I did an REU, which stands for Research Experiences for Undergrads. These are National Science Foundation funded internships where they take undergrads from one university, send them to another one to spend 10 weeks. So I got to come to Hatfield. This is my first time in Oregon, very exciting. And I worked in Sarah Henkel's lab, and I focused on shrimp. So studied a lot of different things. I spent a lot of time working on prayon, which is a really common shrimp that we have here in Oregon. And I spent like endless hours in the lab separating out shrimp and putting them through a bomb calorimeter, which I'll talk about in a little bit what that is. And then I went straight into my master's from undergrad, and I did this at CSU Fullerton, which if you're not sure where Fullerton is, it's right next to Disneyland. So if you've been to Disneyland, you've probably been in Fullerton. And I worked with Jen Bernaford, who also did her PhD at OSU. And this is where I switched into my kelp focus. And I spent a lot of time analyzing kelps and how they impact all the other algae around them. And I did this in Southern California and Northern Washington. So I tend to say that I got an honorary degree from University of Washington because I spent the majority of my time at the Friday Harbor Labs. And then I love school, so just kept going. And I stayed, I came back to OSU, which is very exciting. I came back to Sarah Henkel's lab, who's the same person that I did my internship with when I was an undergrad, because we loved working together so well. And it's very important that you pick a boss that you like working with, right? So I was glad to come back here and continue working with kelp. And I like to show this picture of the Marine Studies building because when I was an intern, this building was not here. This was just like ditch, sand, dirt. And now the new building is here. So whenever we get our new undergrads come in, I'm always like, you have it good. Not everybody had this at one point. So. And some background on how I developed my love for algae. I did not take my first algae class until the very last semester as an undergrad. So I took every other thing related to marine biology. And at the very end, I took my first algae class and was like, this is what I love. So I wanted to stay with botany. And so I ended up taking a marine botany course. After taking that, algae class, and I went to University of Washington to take that for the summer. And then when you go into your master's, you can kind of develop your project. So I picked a kelp that's very common along the West Coast, and I've just kind of kept rolling with that. So marine botany is a good way to take botany and the ocean together. And those are two things that I love. So that's kind of what I stayed with. And so now let's get into what these groups are. So kelps are a type of algae. And a lot of people will ask what are algae, and it's not an easy group to describe. So algae is not one single characteristic. It's not like how we can describe mammals, or there's one unifying characteristic and we can group them all together. Algae are very diverse, and it makes it very hard to categorize them together. They can range in size from single cells to really large seaweeds like kelps. They all do photosynthesize, but they've evolved through multiple pathways, which I'll talk about in a little bit. They are almost entirely aquatic. So those are some of the unifying characteristics, but there's always going to be exceptions to that rule. So algae are all over the place. And for this, there's three main groups that I want you to think of when I talk about algae. There's the greens, the browns, and the reds. So the greens have about 6,000 species described so far, and they have the very distinct color of being green. They're dominated by chlorophyll, so that's why they appear really green to us. And then there's the brown class. They have about 2,000 species described so far, and they appear very brown to us because of their pigments. And then the final group is reds. They are the most abundant so far. There's about 7,000 species described, and they do have that dominating red pigment that we see. And so kelps are a brown algae. They fall into this class of Phaeophyceae, and they do have this like brown color to us. This is one of my 450 students at Hatfield holding up the classic bull kelp that we have here in Oregon. So the kelps definitely get the biggest of all the algae that we have. And then this is just a graph to show you what I mean by these three different groups. So we have algae, which I said is the largest group. A lot of things fit into that category of being an algae. And then there's seaweeds, and then there's kelp. So all kelps are seaweeds, but not all seaweeds are kelps. So like if you see greens, those are seaweeds, but they're not kelps. And if you see reds, those are seaweeds, but they're not kelp. So now a question for you guys. Are all algae considered plants? Put a thumbs up if you think yes, and thumbs down if you think no, and then sideways if you think this is a trick. This is a trick. So first I'll ask you, what is the definition of a plant? So I looked up, I literally asked ChatGPT, what does it think a plant is? And it told me a living organism that absorbs water and, some type of inorganic substances through its roots, and it photosynthesizes. That seems like a classic definition of what we use for plants. Whereas the botanist definition is a little bit more nuanced and more jargony. It's described as a living organism that is a product of primary endosymbiosis. Yeah, what is that? So endosymbiosis is just this concept where one photosynthesizing cell was engulfed by another one. And we know this because if you learned like way back in Bio 101, plants have two cell membranes, which human beings, we just have one in our cells. And the reason for that is because you can see this tiny little picture. There was one photosynthesizing organism already with a membrane. It got engulfed by another one. So then it ended up with two. And all plants have these two membranes. So true plants fall in this group right here. It's all of our land plants that we have here terrestrially. And then red algae and green algae also fall in here. But browns have three membranes. They've gone through this process twice, so we don't count them as true plants. So it's a trick question. Greens and reds are considered true plants. Browns are not considered true plants, but I tended to talk about them as plants. That's totally okay. I won't mark you down if you say it's a plant. And then here's just a picture to show how these groups are all kind of separated out in terms of like the tree of life for a lot of things that photosynthesize. You can see over here on this green branch, here's all of our land plants, and then here's the red algae and the green algae. So they're really close together. They've gone through that endosymbiosis kind of at the same time, whereas the brown algae separate out pretty substantially. So just how they separate out. Okay, next question for you guys. Are kelps found globally? Yeah. And if so, where else have you seen them? Or do you know that they exist?
Temperate climates.
Temperate climates. Yeah, perfect answer. So here's a map, like a loose map of where our major kelp forests are. So the green lines indicate the coastlines that they occupy. So you can see we are dominated by them here in on the Pacific Northwest and the west coast of North America. Also, Southern America has a lot, South America, and then all of the southern areas of the like southern hemisphere have a lot. And then we have some. Japan has a lot of big kelp forests, and then some of the European areas do as well. But there's nothing in the middle. Why? What's in the middle?
Land.
Land? Yes.
Deep.
The line that runs around the world and that's in the warmest area, that's the middle would be the.
Yes, the middle is the warmest. Do you know what's in the ocean there instead? I'll give you a hint. Australia is commonly associated with it. Coral reefs. Yeah. So here's just a picture of the coral reefs that dominate the world. So the Caribbean has a lot. Of course, the Western Pacific has a lot, and then more over into the Pacific on our side. So the middle, the warmest areas, the equator, the tropics are dominated by coral reefs. You don't see them conflicting with each other. So you're either going to have dominating kelp forests or dominating coral reefs, but you usually don't see them interacting. And that's because of upwelling. So we have a system here in Oregon. We're known for having very strong upwelling, and upwelling is just a process where the wind pushes off all the water away from us, and that leaves space for new cold, deep water to come up. And it's usually very nutrient rich. It's also really cold, and those are things that kelps do really well at. Those are good conditions for them. So all the zones where we have a lot of upwelling also coincide with our big kelp forests. So you'll see there's like about 4 major upwelling systems or globally. We have a really big one here in the Pacific Northwest, and then there's one in Chile, and then there's 2 other ones that also coincide with where the kelp forests are. So usually these things go hand in hand. Yeah. So now I'm going to go into some quick ecology background and how it impacts the communities of seaweeds, and just some quick terminology here. I'm going to talk a lot about biotic and abiotic. For biotic, these are living interactions. So that means they're 2 living things that are interacting with each other, whereas abiotic means without life. So those are the non-living interactions. So an example here, like of a biotic interaction could be competition, competing for the same thing with another living organism, whereas abiotic could be sunlight impacting them, or space, or waves, things like that. So I'm going to have you take a look at this picture. This is Boiler Bay up in Depot Bay, and I want you to try to think about how many abiotic and biotic factors that could possibly be impacting seaweeds here. If you think of any, call them out.
Biotic.
Abiotic and biotic. Tides, great. Heat, temperature, good. Say it again. The square net thingies.
Scientists.
Yeah, scientists. Yeah, human beings. The square nets. Yeah, you're right. Competition. That's a great one.
Erosion.
Erosion. Great. Pollution. Nutrients. Great. Yeah, so some of the abiotic ones, temperatures are a really big one. And when you're exposed intertidally like this, which means you get exposed when the tide goes out, you have air temperature and water temperature. So you have 2 different conflicting temperatures impacting you. Light is a really big one. pH is another one, what the pH is of the water. Salinity, how salty it is. Wave motion is a big one. Nutrients, those are just some of the possible ones that impact them. And then for biotic, definitely space competition is a big one. As you can see, every single inch of this place is covered by seaweeds. So they are definitely competing for these space. They overlap. You'll see a bunch like on top of each other. Predation is another big one that I didn't hear yet. And then human trampling. Of course, humans influence everything they touch, right? And then this is just a good picture to show you how dynamic Boiler Bay is and the entire coast of Oregon, right? This is the exact same site. This is me standing out here because I got trapped when the tides came in and I just had to wait for them to go back out before I could cross here. And this is the exact same shelf. So I'm just a little bit off to the side. So you can see how variable the waves are, the variable how the light is, and how these organisms experience very different conditions depending on the day. So just to get into these conditions, for light and shading, this is always described as a love-hate relationship, very similar to houseplants. If you give them too much light, they might wilt or change their pigments. If you give them too little, they might start to droop. So all algae and all photosynthesizing organisms have varying different light tolerances, right? So not all of them are the same, even within each group, those three groups that I described, the reds, the greens, and the browns, each species prefers something different, which makes it really hard to protect them. So you'll see that later. And here's a good example of just where you are globally, like how the tropics, right? If you're in the middle, you're going to experience very different temperatures and very different light input than if you were somebody else, like in the temperate zone, like here. The time of day matters, how cloudy it gets. That's really influential in Oregon, and even the angle that the sun is hitting us based on the season, right? And then this is just a quick graph to show you. This is one of our invasive species here in Oregon. It's called sargassum. It is really pretty though, so I don't mind it. And this is just a study where they wanted to see if can you keep adding light, and will they just keep photosynthesizing more, or does it have some kind of threshold? And you can see by this figure here that around this level of irradiance, they were increasing their photosynthesis, but then it just plateaus. So photosynthesis has a ceiling, and this is true of almost all photosynthesizing organisms. You can keep adding light to them. That doesn't mean they're going to keep photosynthesizing even more, right? Everybody has a threshold. So this is where that comes in with, like, too much light can actually be a bad thing, or it won't benefit them in any way. And then what's important to think about too is a lot of these variables don't act alone. Water levels influence light levels, right? And we have two big swings in our tides here in Oregon. So you'll see big high tide and low tide shifts. And so now I have a question for you guys. Do you think kelps photosynthesize the most when they're submerged during high tide, or when they're exposed during low tide, or somewhere in the middle?
High tide.
And this is an example here of them being exposed during low tide here on the right. Somewhere in the middle. It's actually during high tide, which is pretty surprising. That's the opposite of terrestrial plants, right? So yeah, most seaweeds do photosynthesize the most when they are fully submerged in water. And this is just a graph to show, this was one species that they worked with. This is the species up here, which we also have here in Oregon, that shows that they performed the best when they were at the highest tide, so fully submerged. And then for temperature, temperature is really important because, as I mentioned, thermal tolerance varies by each species. It can even vary by individual, which gets real complicated. And if temperatures are too high, right, that can damage cellular structures and can lead to death. But if temperatures are too low, that also can damage cellular structures and can lead to things like ice crystallization, which I've seen in some of the low tides up in Washington, where it gets real cold when the low tides are happening at the wrong time. And this is just a picture of, I took this screen grab from 2 days ago for NOAA. So this is what our temperatures are sitting at right now off of our coast. And you can see that most of the sea surface temperatures right off of Oregon are sitting at about 10 degrees Celsius. And then as you start to go north, you start to get even colder. And as you start to get down into California, you start to go up to like 12 or 13 degrees Celsius. But Oregon usually is sustained at around 10 degrees Celsius. And then for pH, this is the big one that people talk about because ocean acidification is a very big problem for things that calcify. And so as we add CO2 to the atmosphere, it's going to lead to more acidic ocean waters, as you can see by this graph right here with the ocean pH. And surprisingly, a lot of algae actually do calcify, like animals that have shells. So there's some species of algae like this that are really hard, they have a hard exterior, and they do use calcium carbonate to make the like outer skeleton that they have. And surprisingly, a lot of studies have shown that more acidic waters are actually beneficial to them. They're actually going to do better, and they're going to be able to calcify even better. So pretty interesting. And then I'll just briefly touch on one of the biotic interactions for herbivory, because this is the big one that's influencing where we are currently with our kelp forests. This is important because a lot of creatures in the ocean like to eat seaweed. For instance, we have crabs that are commonly called kelp crabs because they hang out solely on kelp, and that's what they like to eat. And then, of course, we have other types of scavengers like purple urchins that can just mow down whole fields of seaweeds because they really like to eat it. And then this up here is a picture of one of the seaweeds that I had in the lab. And this is being eaten by things like snails and limpets that can use their little scraper. They have like little scraper tongues, and they can just go along real slowly and like rip apart kelps. And surprisingly, they all really have preferences, and like really strong preferences. So you can give them, there's a lot of studies where you can put like an urchin in the center and give it 20 different species of kelp, and it will pick one every single time. So the preferences of these herbivores really matter because they can mow down and destroy entire kelp beds if it's what they prefer to eat. So for the current outlooks, also going to talk about the current losers. You're going to see you see a lot of headlines like this, like the vanishing forest, or big headlines like 90% of kelp forest gone. And this is happening. This is not great. One of the first studies that came out, I think this came out in 2019, and this came from some big schools like Santa Cruz and UC Davis and UC Santa Barbara. They were all coming together to kind of analyze as much long-term data that they could for kelp forests. And you can see here that in 2008, this is for Northern California. For 2008, all this green is like aerial photographs that they analyzed to see how much kelp forest is like floating on the surface. So 2008 looks pretty good, nice and healthy. 2014, you see a lot of it disappear. And then 2015 and 2016, it's almost gone completely from some counties and just few left behind in some places. So this is huge losses starting around 2014. And then here, I'm just going to be talking about bull kelp because that's the most common one that we have in Oregon, really common in California too, but there is the other species, the giant kelp, which we have a little bit here in Oregon, but I'm going to focus on the bull kelp today. And then what's happening in Oregon? So there's a great group called ORCA, which stands for the Oregon Kelp Alliance. And they've used a lot of different techniques to calculate how bull kelp canopy has changed from 2010 to 2022. And they just put out an amazing report this year that includes all this data. It's a really nice long report. If you're interested in that kind of stuff, it's available online for free. But essentially what they did is that you can see here, we're in Newport here, they have looked at they either used satellite footage for these central sites, or they used like helicopter footage to analyze these southern sites. And what they found was that basically anything that looks red had huge losses from 2010 to 2022. Like Cape Lookout lost almost 92% of its canopy. Cape Foulweather has lost 44%. And then the bigger Cape, like Cape Blanco, has lost 95%. Brookings is down 74%. And then there are only two positives. Rogue Reef had 1% gain. That's good. And then Cape Argo had 23% gain. But overall, huge losses of our bull kelp canopy here in Oregon. So what does this actually look like from the surface, right? So these are some pictures from the same paper that I just described, where you can see this is like a healthy canopy covering the surface. So very hard to get through. If you're a diver, you know how complicated it is to try to get through these canopies. So you have a lot of canopy covering that surface, and then underneath it usually creates some kind of nursery ground. So you're going to get a lot of other diversity of other types of algae underneath it. And then you'll start to see other things like a lot of abalone and other organisms underneath that. When you remove the canopy or the canopy disappears, you start to get barrens because you're lacking the protection of the upper canopy. And if too much light penetrates this area, then these cannot handle it because these prefer kind of lower light levels. So they'll disappear, and then you'll start to see like this is all crustose, coralline algae, that will also disappear because it can't handle too much light impacting it. So you start to get things like this where urchins are just dominating everything. So what are the mechanisms that are driving the losses? Of course, as I've mentioned, it's not usually just one thing that's causing this to happen. It's a cascading or combined effect of many things that are coming together. The first big one is temperature. Of course, our oceans are heating up. Like I mentioned, Oregon usually sits around 10 degrees Celsius. And in 2014, our average sea surface temperature for the Pacific Northwest was like 14, 15 degrees Celsius. So it jumped pretty significantly, and this was impacting a lot of places. So the temperature jumped. And then, as I mentioned, we had sea star wasting disease. We lost a lot of stars. And I mean like 90 to 95% of our stars just like melted on themselves and disappeared. And the biggest problem for us was we lost the sunflower star. And the sunflower star, beautiful stars, they have up to 25 arms. If you've ever been to the Oregon Coast Aquarium, they have really nice ones there that are still living and will hopefully continue to live, that are really giant. But we lost almost all of them. In California, there are none. Here in Oregon, we're starting to see little babies. And like to get to this size takes them almost 5 years just to grow to that size. So when you're getting the massive ones, you know they're old. They're going to take a long time to get there. So we started to see some little ones here in Oregon, especially in Newport, like under the bridge. People have been reporting them, which is really good to see them. But we need the big adults because the big adults are what can eat urchins. So if you look at these graphs, you can see here that through the years, as we lost our sunflower stars here, we started to gain a lot of urchins. So now, because the urchins don't have predators, of course, we don't have otters here either. So urchins are just like free growing and they are just populating as much space as they possibly can. So all of these effects combined have really led to why we're losing so much kelp. And then for bull kelp specifically, there has been some studies done to see, what is its thermal tolerance. And by this graph, this is just a graph analyzing, they looked at how well it grows under different temperatures. And then they did different ranges from 5 degrees Celsius all the way up to 22 degrees Celsius. And you can see that the peak of its growth is around 12 or 13 degrees Celsius. So that's when it's performing the best. But once you get above 13 degrees Celsius, it starts to drop. So that means it's hit its thermal tolerance and now it's going to start not growing as well because it's starting to get stressed out. So definitely has an upper thermal tolerance of about 13 degrees Celsius. So when we get above that, now it's got many things happening. The temperature is too high, so it's going to have some cellular stress. And then now we've got urchins everywhere and they're just going to mow it down because it becomes even easier to eat when it gets cellularly stressed out. So not great. All these things combined is definitely leading to a loss of our bull kelp. But what about other kelps? So today I'm going to talk to you about feather boa kelp. This is the one I studied. This is me holding one of them. They can grow just as long as bull kelp, but they don't make the big, pretty kelp forest beds. So I think that's why they're a little underappreciated. But they have a cool, unique structure where with bull kelp, as I showed here, it's got the one big bulb on it, which helps it stay upright when it's submerged. Whereas Egregia, or the feather boa kelp, has many little tiny ones throughout the entire length of it. So instead of having one big bulb, it just has tiny little ones, all filled with air that help it stay upright. So pretty unique structure. So its scientific name is Egregia menziesii, and I'm just going to be referring to it as Egregia for the rest of this talk. It is considered a foundation species, which means it dominates the areas that it occurs in. So it occurs, especially in Oregon, in the intertidal, and it can cover whole areas. And it is one of the largest, most common brown seaweeds on the West Coast. It's also perennial, which means it can live for many years, which is different from bull kelp. Bull kelp usually go through an annual cycle, so that's why you'll see them appear and then they'll disappear. And then next year they'll appear again and then they'll disappear. So Egregia is here year-round all the time. And then it also is unique because it has a really large geographic range. Egregia is found all the way from Alaska down to Baja California, whereas bull kelp has a different range. It's more limited. You won't see bull kelp start to get into the Baja area. That's a little too warm for it, whereas Egregia does occur down there. So for my studies, I'm going to go into the first objective here, which I really wanted just to examine the seasonal variation and the effects of heat waves that we've been having here in Oregon on the nutritional value of Egregia. Because a lot of things like eat Egregia, and I want to know how how the calories change seasonally. So for just quick methods, I've been collecting Egregia from both Boiler Bay and Seal Rock since January 2023, and I did this all the way through November of last year, and I collected about 100 individuals over the course of those 2 years. And then I did something called bond calorimetry, which is this amazing, like kind of old style way to analyze nutritional value, but it's a lot of fun for me because you can take a dried sample. So you dry kelp like in an oven, and then you smash it all up and you can put it in here. And what happens is you ignite the kelp in a very pressurized bomb system, essentially. And when it burns, it will release energy, heat up the water around it, and then I can calculate that temperature change for how much energy came out of the sample. So that's a way to check to see how much energy is coming out of these kelps when they burn. A lot of fun. And just a quick note, I started this project right in January 2023, and then we did have a heat wave occur here in Oregon in August of 2023. So this is the kind of report that came out from NOAA in July, like July 30th, where they said, you know, we're starting to see really anomalous high temperatures on our coastlines. So yeah, so now I can compare my data to that heat wave. So what I'm going to show here is these are the calories, or how calorically dense the samples are. And then here on the bottom is just the first year of data. So I took samples in January, May, August, September, and November of 2023. And then as a reminder, the heat wave was right here in August of 2023. And the main takeaway here is that calories were not impacted by heat waves. Not huge differences in the caloric content of the algae. Even when there was heat waves happening, even when they were supposed to be stressed out by these temperatures, their calories stayed pretty stable. And then when you add the second year of data, they stay even more stable, if not increased just a little bit. But overall, the calories of this species, very stable. Stable year round, seasonally, stable species. So this is good. It indicates that temperature does not really influence their nutritional content, which is good for other organisms that rely on them as a food source. And then for my second objective, I'm looking at the consequences of increased temperatures on the physiology of Egregia. And so what I mean there is I'm really looking at the thermal tolerance of this species to see if it's better than the other ones that I talked about earlier. So I've been collecting juveniles of Egregia. So Egregia can get really long, and I've just been getting the ones that are like arm length, like less than a meter long. And that's because they need to be able to fit into these bottles. So I'm constrained by size. And for the study that I'm going to show you today, I exposed them to one of three temperatures. So I either exposed them to 13 degrees, 14 degrees, or 15 degrees Celsius. And then this is what it looks like at Hatfield. They're each given their own little tank, and then the water kind of recirculates. And they have like bubblers in there that keep them tumbling and happy with their lights. And then I track their physiology via dissolved oxygen, because that's kind of like an indicator of how well they're going through photosynthesis. So how much dissolved oxygen do they put out when they're photosynthesizing, and how much dissolved oxygen do they take up when they're going through respiration? So for the 2024 experiment, this graph is a lot, but what I want you to take in is that I'm just talking about gross photosynthesis. So this is overall output of their photosynthetic rates. And then the x-axis here is day. So I did this experiment for 24 days. So that's 24 days of sustained exposure to these temperatures. And then basically the main color code here is that ambient, or 13 degrees Celsius, is the lowest temperature. Orange indicates kind of a warmer middle temperature of 14 degrees Celsius. And then the red is supposed to be the warmest of 15 degrees Celsius, which we don't really experience here in Oregon. So this should be like very stressful to them, hypothetically. But you can see by this graph that for days 0, 6, and 12, there's no big differences, but you do start to see the colors separate out. And by day 18 and 24, the red group, which is the highest temperature, is performing the best. So they are photosynthesizing the best at the highest temperatures. Whereas the lower temperature, like 13 degrees Celsius, had a reduced photosynthetic ability. So overall, for this species, warmer water temperatures does lead to more photosynthesis for them. And then this does impact their calories. So I took all these samples at the end of the experiment and I put them through the bomb calorimeter. And you can see that the caloric density of these species for the warmest group was also highest in the ones that were experiencing the highest temperatures. And the two for the ambient and the warm, they had reduced caloric density. So they actually dropped their calories a little bit. And how does this compare to the graph that I showed you before in terms of like what's the average of this species? The average for calories in the ones that I did not manipulate, I just took straight from the ocean, they average about 15,500 calories. And so the red group is sitting about there, just a little bit higher. So just basically maintaining the caloric density that we see out in the ocean, whereas the ones that were exposed to lower temperatures dropped some calories, which is interesting. And you might be asking, did I ever go higher than 15 degrees Celsius? I did, but unfortunately they die. So any temperatures above 15 degrees Celsius, I did this experiment in 2023 or yeah, 2 years ago. And I lost 70% of my individuals. So 15 degrees Celsius is their thermal max, which is really interesting because once you expose them to 16 or 18 by day 14, I had lost over half of those individuals. So they definitely have a tolerance. 15 degrees Celsius seems to be their better performing temperature, but above that is bad and below that is bad, right? So overall, Egregia does seem like it will be a winner for the future because it does perform the best at 15 degrees Celsius. Anything below or above that did lead to some reduced photosynthetic rates. And it does have a much higher thermal tolerance compared to other kelps, like the bull kelp that max at about 12, 13 degrees Celsius. So Egregia has a little bit more space in terms of ocean warming that it can be sustained at. So it will be hopefully a winner and we'll be able to fill a space. If we lose too much bull kelp, Egregia can hopefully be a redundant species there. One more second. And then just some tips for what I've been doing to make me feel not all doom and gloom about all of our kelp being lost. The first one is to appreciate the rack, which is all of the stuff that appears on the beaches. It's ugly and it's smelly and it takes up a lot of space. But if you see a lot of rack, then it means our kelp forests are doing really good, right? It means we're getting, it means we're getting a lot of kelp. They're going through their annual cycling. If they end up over here, that means they've gone through their life cycle and they're doing good. If you see empty beaches all the time, then that means there's nothing to replenish the future years, right? So you want to see rack. So if you see it on the beach, say, you're doing good, walk away from it. A second tip is to definitely volunteer. I volunteer both at the Oregon Coast Aquarium as a diver and at the Hatfield Marine Science Center. And it's always fun to talk to people about this kind of stuff. Because people get real jazzed, right, about hearing about kelp and how things are going. And of course, when you're in places like that, like the Oregon Coast Aquarium and the Hatfield Marine Science Center, they're keeping a lot of the species that are tending to go extinct in Oregon. They have, right? So it's nice to see them, still see representatives that can hopefully help the population later. And then another big one is to eat kelp products, which surprisingly, there's a big industry for kelp now, especially in Alaska. It's a big aquaculture industry. And the reason I say to eat kelp products is because higher profits for those industries leads to more like product protection by those industries, right? Because they need the populations outside to sustain their populations that they're farming. So one of the companies that I like is Barnacle Foods. They make a lot of kelp related salsas and things like that that actually taste very good. So if you've never tried kelp, this is your chance. And then the last tip is to just definitely advocate, vote for protections and spending to protect kelp forests. So like big, the state of Oregon has been supporting a lot of kelp forest action, especially through Orca. So they've been facilitating a lot of research that way, which is good. We wouldn't have a lot of this data without state funding. And then my last plug is to join us at Hatfield. We have our Hatfield Marine Science Day coming up next week. It's on Saturday. It's from 10 a.m. to 4 p.m. It's free to the public. I'll be there. I'll have a little algae cyanotype station that I'm working on. So definitely come to Hatfield next week if you're around. I'll take questions now.
I just wanted to mention for some of us who are not Celsius Fahrenheit, 15 degrees Celsius is 59 degrees Fahrenheit.
Yes. For the audience on Zoom, 15 degrees Celsius is 59 degrees Fahrenheit. Yes.
Yeah, I grew up on the East Coast of Long Island,
Mhm.
and I traveled all the way up to Maritimes,
Okay.
Prince Edward Island, Halifax.
Mhm.
I've never seen kelp anywhere on the East Coast.
Mhm.
It showed a little bit, and I'm curious as to where.
Yeah, so there is some representatives that fall within the family. I think the most common one, which isn't necessarily a kelp, but it is a brown algae, is the Ascophyllum genus, which is still creates pretty big mats. I've definitely seen it, like when I was around New Hampshire, it covers the places there.
Beach in New Hampshire.
Yeah, you're right. But yeah, there are some that definitely occur there, but nothing compared to what we have on the West Coast, right? Just in terms of diversity. But yeah.
And then I noticed recently, I volunteered at the aquarium.
Okay, very nice.
I noticed kelp at Depot Bay and then at Boiler Beach, and it's noticeable. Is the action of the waves, does that keep the urchins from eating all of the kelp in those two spots?
So the question is, does the action around Depot Bay and Boiler Bay keep the urchins from eating the kelps that are there? So there's natural cycling of the life cycles of kelp. So they tend to appear in the spring, like the big adults start to appear in the spring, because there's like a microscopic phase that usually like saves and is, kept kind of microscopic in the wintertime, and it waits until the light starts to get good, and that's when it develops into the big adults. So that's why you're seeing it starting now. And then wave action does keep urchins from moving around, which we've seen, like if you put them in the lab and you expose them to a lot, they'll just kind of like stay in the little crevices that they make, because they can like dig out holes. But that doesn't mean that they're not going to chew it down at some point. It just kind of depends on how abundant the kelp forest can get, and if it's big enough that the population won't chew it down. But it's really dependent on the conditions. But yeah. Back.
I'm just curious how long it takes kelp to grow from very small.
The question is, how long does it take kelp to grow from very small to very large? So the microscopic phase, it's called a gametophyte. Actually, like, let's see, can I show my slide here? I think I don't have it in here, but there's best way to think about every algae has a different life cycle. So it makes it, of course, very complicated, but for kelps, they have the full adult, right? Which looks like a very obvious big adult to us. And then they have a microscopic phase, which is where you see the separation of the sexes. So you see like the two sexes, and they stay microscopic. And then they release gametes out into the water column. The gametes come together, that settles again, and then that is what becomes the adult. So they have to go through these like multiple phase situation. But for like for bull kelp, for instance, I think it's just like a couple months. They can grow very fast. So they're like really big, long, with all the fronds. That's just like maybe 2 months, because their life cycles are very fast. And Egregia definitely doesn't grow as fast as that. It can get just as long, but it takes like many years to get to the same growth speed that the big bull kelps do. Yes.
Can you speak to this issue of urchin barons
Mhm.
and that food web?
Yeah, so
Uh,
the question was to talk a little bit more about the urchin kind of cascading barons and how we get there and how the food web is interacting. So is that what I heard?
Yeah.
Okay. Well, okay, so first I'll start off by saying food webs are extremely complicated. We tend to talk about them at kind of like 2 or 3 levels, but it's usually like 30 different levels that are feeding into the same food web. So it gets real complicated. But the best one to think about, the one that I like to describe the most, is the one where it's like urchins related to otters related to kelp is like the classic one that we've used. And so of course, when you have otters, they eat urchins no problem. They like to crack them on their bellies and they have a great time. And that has an indirect effect. When you have a lot of otters, you have really healthy kelp forests. So when you lose your otters, urchin populations get out of control, and then it has that cascading effect onto the kind of the forest floor after that. And they just mow down everything. But there are a lot of other things that can keep urchins in check. We haven't had otters in Oregon for a really long time. So that wasn't like a new thing for our food web. It was really when we lost basically their only predator here in Oregon, which was the big sunflower star, which can mow down like hundreds of urchins in like less than a month. It's very quick because they get so large. So when we lost them, that's when we saw the same effect that like if we had lost sea otters. Those were like essentially the same thing. Losing sea otters, losing sunflower stars has the same effect on the urchin population getting out of control. So yeah, and some other things do eat urchins, like birds. If they're exposed, especially in the intertidal, I'll see birds kind of ripping them up. And then humans. I mean, we are also part of the trophic cascade because humans go into these areas and will chisel out urchins, right? And you can eat them at home. And we do have like a food industry related to uni. So we do definitely have a similar cascading effect on them. So that is a big movement right now. Like Orca's been working with divers to kind of smash as many urchins as they possibly can or collect as many urchins as they possibly can. It just depends on the permits and the sites that they're trying to do it at. But they're trying to alleviate the urchin population. So that's the way that like humans impact the cascading effect. And in California, they're doing something very similar. They've been doing longer studies than us, where they have taken out a bunch. The divers just go on and just kind of like scoop them into bags as they're swimming along. And the kelp forests are doing a little better. They're starting to see the spore fights grow up into adults, which is good. But the urchins are still definitely like they're growing at such a rate that it would be like impossible for us to take them out, like the whole area, because they just can move in so quick, right? And we can't breathe underwater, so we're time limited too. Yeah.
So has there been any definite answers to what causes sea star wasting?
The question is, has there been any definite answers to what caused sea star wasting disease? And yes, there are a lot of people who study marine disease. Right now, so it was like when it first came out, people were trying to figure out what it was. It also came out at the same time we were having a marine heat wave. So a lot of people assume it's like temperature related too, which it definitely is. But it was like a virus, a denzovirus that came in, and it had always been present in the water, but I think the increased temperature exposed the sea stars physiologically to being disturbed by the virus. And there's been some studies now that are connecting it to like the mad cow disease and things like that. It has a very similar structure to that. So somehow that is just very abundant in the seawater. And if the sea stars get stressed out, it kind of exposes them to disease, which is similar to us. You know, if we get really stressed out or if we're immunocompromised and then we get exposed to something, it has like even more of an effect, right, than it normally would. So very similar to what happened with sea stars. And some species were way more exposed than others. So like, for instance, the sunflower star got decimated. Pisaster, which is the common like purple one that we have here in Oregon, that one lost a lot, but it has rebounded. So you can definitely find them a lot. The site where I see them the most is Bob Creek. They cover Bob Creek now, which is really surprising. I don't know what they're all doing there, but at least I'm happy to see them. And then other species like a common one that I really like is the leather sea star, which doesn't have spines. It doesn't have any armor. It's just really soft. That one didn't really have any wasting. So it just seemed kind of more robust to the effects of sea star wasting. So just some species.
The leather, is that in Oregon?
Yeah, we have it in Oregon, but it's usually subtidal. So every once in a while you'll see it in the intertidal.
That's more weather.
We definitely have it, yeah. But yeah, it's very similar to bat stars too. If you've seen, like we have big bat stars at the Oregon Coast Aquarium too. So
It seemed like the suit.
yeah. Yes.
Yeah, you mentioned something about invasive species, issue.
So the question is about invasive species and if that's a really big issue. So yeah, I'll pull up the picture that I had. I think it's pretty. This one, yeah, sargassum. I bring it up because for a lot of the like organisms or organizations that I dive for, like Reef Check, we monitor invasives, so it's always something that you're like, if you see it, you have to like make sure that you're monitoring that you see it. This is an invasive, and the term invasive means that it's not originally from here, right? So it's moved from somewhere else. But that doesn't also mean that it has a bad negative effect. I think we tend to assume invasives are doing bad things, but that's not always the case. For instance, this species sargassum is from Japan originally, so it just kind of like moved across the Bering Sea down, and now it's kind of just like covering the entire West Coast. But it's not like out competing other species that I've seen. So like Egregia definitely does not compete with this species. But and it also doesn't grow as big, but it's also been here for almost 100 years. So at what point do we decide that it's no longer an invasive? And that's like that is a huge, that's up for debate, right? Scientists debate about this all the time, is like, when does a species that's been here for so long just become like a naturalized species if it's embedded into the community? So like a species like this, I would describe it as naturalized. It isn't invasive, but it's been here a really long time. It doesn't seem to be like removing anybody or having a negative effect. Kelp crabs like to eat it, so it's kind of just like here. And it's not like we can rip it all out. That would be impossible too. So it's kind of like we just have to evaluate things as they move forward. But the biggest thing is like making sure, I think the most important thing is to just try not to introduce any new ones, right? This was probably introduced by ballast water or something like that from big ships before we realized what we had been doing and moving water from different parts of the Pacific into other parts of the Pacific. And then another way that we get invasives, which I always like to talk about, is with tsunamis. So like we had a relatively big tsunami right in Japan, like 2 decades ago, or roughly around that time. And we started getting debris from that part of the world, and they brought a lot of invasives with them. So if you don't like extract that or like burn off those pieces, then they will settle and become part of our community. And that's just something you kind of have to monitor. It's not necessarily something that you can just like rip all of them out. So, but it hasn't been a huge problem for us. I would love if like more kelp came in and tried to fill where we're losing kelp. Yeah.
All of the kelps, into rocks like the bull kelp does?
Yes, so all of the kelps do. So let me see if I have any pictures. But kelps and almost all algae have something called like a holdfast, right? Instead of roots. So they're not like embedded into the ground. They just need to attach to some kind of hard surface. And yeah, most of them attach to rocks. And that's why also we tend to see kelps on like big rocky reefs, right? Instead of like sandy beaches. So I like to, when I forgot to mention for the orca study here, right? They didn't analyze any of this stuff in the middle because we don't have kelps there, because that's all sandy beach. So we don't have any just like natural rocky reef out here, so we don't get kelp forests out there. Whereas off of these capes, we have a lot of rock substance, surface going off of these capes. So that creates the habitat for them. So yeah, they definitely need some type of rocky surface. And I have seen that like, because if you've noticed in Oregon, we have a lot of sand movement. Sand comes in and out, right? So if there's still rock underneath, if they get buried a little bit, they're usually totally okay because they're still secured to the rock underneath, and they can be buried in like feet of sand. As long as there's rock under there, they're okay. But if it's just sand for like 10 or 20 feet underneath them, then there's nothing for them to attach to. So, yeah.
Can you speak about eelgrass meadows?
Yeah, the question was, can I speak about eelgrass meadows? Yes. So I'll come back next year and talk about the study that I'm doing right now. I'm doing the exact same study with the temperature stuff, with the bottles, but I am using surfgrass and Egregia, because these two things, the seagrasses and the kelps, can co-occur in the intertidal. So you'll see them overlapping each other. And surfgrass, also the seagrasses also have really large geographic ranges, right? They can be found all the way from Alaska to Baja. So that implies that they have a really good thermal tolerance, but no one's really investigated it here in Oregon. So I'm looking to see like, if we end up with just these two species, if we end up with just Egregia and just a bunch of seagrass, like who's going to be the winner now between those two? So that's what I'm working on right now. I'll report back. But for in Oregon, we have some invasives also of seagrasses. So there's, I didn't go into this because, seagrasses are true plants, they're not algae. But I can briefly talk about it. We have, Zostera is our species that we have in estuaries. So that's kind of like the one that you're most commonly gonna see in the bays, like we have in here. But then we have an invasive too. So we have two Zosteras. We have the Marina, which is like the species that's supposed to be from here. And then we have Japonica because it's from Japan. But they occur in very different zones. So like if you walked the estuary, you would see Marina here and Zostera or Japonica right above it. So they occupy totally different spaces. So people are just kind of tracking how that's influencing the communities and the sand and the bed and the mud and stuff like that. But right now it's not out competing the like current species, if that makes sense. It just kind of came in and was like, okay, well, you're here. So I think I'll just like take up house here. And people have been monitoring that. But in other spaces like San Diego, this is a similar problem. Their species are overlapping. So now you're starting to see competition between those two. So it kind of just depends on where you are and where they have moved into.
What was in that space that Japan?
Nothing. Yeah, there was nothing before. It was just mud and shrimp. Shrimp in the mud. Okay, yes.
So has there been an analysis of sea level rise and potential rocky habitat? How much, the sea level rise and comes onto shore? What the change in rocky silt?
Yeah, so the question was, has there been an analysis on sea level rise and how that influences kind of algae and related to like their structures and if they're running out of space kind of scenario? The biggest problem is like the biggest places that are going to have the biggest impacts is area where you're running into walls. So like when you have sea walls or like kind of like Boiler Bay or Depot Bay, there's nowhere to go, right? It's like you hit rock and then you hit cliff and then now you're nothing. Whereas other places might have just like beach for days. And so that keeps more space open when the sea level comes up. But in Rocky, the biggest concern is species that aren't used to being submerged all the time. So as you bring sea level rise up, right, then you're starting to get everything moved down where they currently were and the sea level's coming up here. So then if you're a species that's used to like more light and then you're getting pushed even deeper, then you're not gonna be able to photosynthesize as well because you're losing your light source. And that's I think the biggest problem is gonna be competition. It's really gonna be like which species can do the best there, and then those will just take over. And other things with sea level rise is the subtidal species, like we see with bull kelp, they are used to certain conditions. They like their certain conditions. They're specifically at their temperatures. They want a perfect thermocline. And when those things change, they do not do well, right? So as you shift the sea level rise, they're also gonna not be able to adapt because they're so used to the conditions that they've been at for thousands of years that now it's like, what do you mean? Can't change? I can't do this. So, yeah.
Scenarios and how that can.
Yeah, so the question was, can I talk a little bit about expected sea surface temperatures and how that's going to impact the thermal tolerances of kelps? So the biggest reports, right, are like the big global ones, which are saying, I think it's like globally, the sea surface temperature everywhere should go up by 1.5 degrees Celsius. But of course, that doesn't sound like a lot, 1.5 degrees Celsius. And that's like the global average of how we should go up, where like when I showed the picture of the marine heat wave here, so this is a common figure where it's showing anomalies in our average temperatures. So the 0 is kind of like what we've averagely been at for like however long this has been monitored, which is like usually at least 100 years. And then if it's deviating, how many degrees is it deviating up and how many is it deviating below? So like for this heat wave, it was like at least 2 or 3 degrees Celsius that we started to heat up. And then we do get the opposite where we start to get colder temperatures. But luckily for kelps, colder temperatures don't impact them. In terms of like losing the populations, they can sustain at below freezing temperatures, which is really interesting. But it's the increasing temperatures that you start to lose populations, like whole populations. So yeah, like 3 degrees Celsius is like definitely becoming the norm now when we have marine heat waves. And then we had our huge marine heat wave in 2014, 2015, which everybody calls the blob. And that was like extreme because of the duration of it. So like my experiments, I really looked at like days and how that affects things.
Floating meaningful.
So I looked at like how the days influence. So like in the first couple of days, I didn't really see any differences. So like the first 12 days, they were all kind of like, okay, this temperature is like fine, but I don't love it. But then after like sustaining those temperatures for long, I start to see big separation in their photosynthetic abilities. And when we had the marine heat wave, the big blob, that was like almost 2 years of like 3 or 4 degrees sustained sea surface temperatures. And so we saw like huge losses of everything. That's when we saw wasting getting the worst. So it's not crazy to assume we're gonna start seeing really temperatures really ramp up when we have marine heat waves, especially coinciding with like El Niños and dry periods. And it's really compared to like the pressure of what's happening on land. So it's gonna get crazy. But everybody predicts the marine heat waves are gonna get more frequent and even longer. So.
Yeah, if we want to end it with this question, which I don't know if anyone else is wondering, but I was wondering, how are the kelp bulbs inflated underwater?
The question is, how are the kelp bulbs inflated underwater? Okay, so kelps just do natural diffusion. So they are just like taking oxygen out of the water and somehow pushing it into the bulbs. It's not weird. So yeah.
Pretty much.
Yeah, pretty much magic. Yeah. And it's also how they, a lot of the nutrients that they take up, they take it straight out of the water, which is very different from terrestrial plants that have to like diffuse it out of the air into their cells. And if you've ever seen like a plant up close, they have little gas structures called stomata, and they open and close, and they look like little kind of lips that are closing. Kelps don't have that. And most algae, almost any algae doesn't have that structure because it can just diffuse over any part of their structure.
Is that the same then for the little bulbs on the Egregia?
Yes, yeah, yeah. They have a lot more of those.